Next, we're going to look at the three parts of the ear: the external or outer ear, middle ear, also known as the tympanic cavity, and the internal or inner ear. The external ear and middle ear are both involved in hearing, whereas the internal ear functions in hearing and equilibrium. There are receptors for hearing and balance located in the internal ear, and they respond to separate stimuli but are activated independently. Next, we're going to look at the structure of the ear. The outer ear is actually called the pinna or auricle. There's different parts of the pinna or auricle. For example, you can name the helix or the lobule, but just the outer ear, make sure that you know that's called the pinna. Then from the pinna to the tympanic membrane, which is also your eardrum, we have a canal, also known as the external acoustic meatus. So from here to here, that is the external acoustic meatus. You can also say that it's the auditory canal. The auditory canal meets with the tympanic membrane, also known as the tympanum. The tympanum is going to separate the external ear from the middle ear. In the middle ear cavity, kind of outline that just a little bit. In the middle ear cavity, we have three bones. Those three bones as a group are called the auditory ossicles. They consist of the malleus, incus, and stapes. So malleus, incus, and stapes. And the stapes sits on top of the oval window. And the oval window is going to be an area that separates the middle ear from the internal ear. The internal ear, or labyrinth, consists of an area of kind of bony carved out structures and then membranous structures that fit in those bony projections. We can see the semicircular canals. There'd be another one over there. And we can see the vestibule. And we can also see a snail shell-like structure called the cochlea. And these are structures found in the internal ear. They're not the only ones. We're going to look at them in more detail, but this is an overview. Only structure that I want to mention before we move forward is the pharyngotympanic or auditory tube. You may also call it the eustachian tube. This is a tube that connects the middle ear to the pharynx, that's your throat. So it's a tube that connects your middle ear to your throat and helps equalize pressure between the two areas. Make sure you know that the pharyngotympanic or auditory tube is located in the middle ear. So external ear structures are as follows: the auricle or pinna with its helix and lobule, the external acoustic meatus, also known as the auditory canal. The tympanic membrane is going to be a barrier between the external ear and the middle ear, and we're going to see it's mainly just connective tissue. You may also know it as your eardrum, but it's able to transfer sound energy or auditory energy to those ossicles that are in the middle ear, those bones. The middle ear, like the outer ear, is air-filled. It's mucosa-lined. It's contained within the temporal bone. It's flanked laterally by the eardrum, immediately by the bony wall, containing both the oval and round windows. The oval windows where the stapes was. The pharyngotympanic or auditory tube is that tube in the middle ear that connects to the pharynx, specifically the nasopharynx, which is the upper level of the throat behind the nasal cavity. And again, it just equalizes pressure in the middle ear cavity with external air pressure. Let's talk more about the middle ear. We have three auditory ossicles or ear ossicles. They're just small bones in the tympanic cavity. They are the malleus, incus, and stapes, and they are suspended in place by ligaments and joined by synovial joints. They do transmit vibratory motion of the eardrum to the oval window, and they have even tensor tympani and stapedius muscles, or muscles that contract reflexively in response to loud sounds in order to prevent damage to those hearing receptors. The point of the auditory ossicles is to amplify auditory signals from the outer ear to prepare them to go into the inner ear. So we amplify auditory signals in the middle ear. We make the amplification or kind of height of the waves that were received from the outer ear bigger. And that's because it's a lot harder for auditory signals or waves to move through fluid than it is through air. And while the outer ear and the middle ear are air-filled, the inner ear is fluid-filled. So again, we're looking at structures of the middle ear, just kind of a review. Here is the tympanum or tympanic membrane, also known as the eardrum. It's going to laterally flank the middle ear. This oval part of the stapes is sitting on the oval window, and that's going to medially flank the middle ear. And here's the round window. Not as obvious. Another structure going to medially flank the middle ear. The auditory ossicles consist of the malleus, incus, and stapes, and they do have other names because of their shape. The hammer, anvil, and stirrup. So the malleus is called the hammer, the incus is called the anvil, and the stapes is called the stirrup. We're going to take another quick look at the inner ear, but a better image would be coming up. And we can see that we have half circles. There's actually 3 of them. These are semicircular canals because they're half circles. They sit atop a chamber called the vestibule. And then you have the snail shell or cochlea. So inside the cochlea is the organ of hearing, whereas the semicircular canals and vestibule, they're going to be concerned more with equilibrium or balance. Other things we can see in this image is the vestibular nerve and the cochlear nerve, which will come together and form the vestibulocochlear nerve, one of your 12 cranial nerves. So here's a better view of the inner ear. Again, there's a bony labyrinth or bony cavities in which the structures of the inner ear sit in. So you have both a bony labyrinth and the membranous labyrinth. Inside the membranes, things like the vestibule, the semicircular canal, and the cochlea, that's where the fluid of the membranous labyrinth would be. So the cochlea contains the organ of Corti. That's the organ of hearing, and that receives sound in the form of vibrations. Stereocilia are kind of tiny hairs of the organ of Corti, are able to convert vibrations to nerve impulses. They act sort of like mechanoreceptors. They're going to distort the membrane of the cells of the organ of Corti. Semicircular canals have receptors that pick up on angular movements of the head. So those are cristoreceptors for the semicircular canals. And then the vestibule has maculae receptors, and they're going to pick up on changes in gravity or changes in the position of the head. You can see that you have an anterior, posterior, and lateral semicircular canal, that the vestibule is actually made up of the utricle and the saccule. And then you can see that there is a canal, this kind of silver one right here, that travels in the center of the cochlea, and that is actually the cochlear duct. Let's look at the vestibule more. So it is an egg-shaped cavity. It's a membranous labyrinth that sits within the bony labyrinth. It has 2 membranous sacs, the saccule and the utricle. The saccule is continuous with the cochlear duct, whereas the utricle was continuous with the semicircular canal. So let's look at their position again. The utricle was superior to the saccule. Saccule again being closest to the cochlea, utricle being closest to the semicircular canals. All right, next we're going to talk about the sacs in detail. For example, we know they have different receptors. They have the maculae, where semicircular canals have the crista ampullae. They're responding to gravity changes and the position of the head. Semicircular canals are responding to angular rotational movements. Here's the receptor. Really want to know the receptor. And you have 3, each are kind of like a half circle or 2/3 of a circle, hence their name, semicircular canals. Together we can say that the utricle, saccule of the vestibule, and the semicircular canals are more concerned with equilibrium or balance, or kind of the position of the head in space, than hearing. They're not concerned with hearing. It's the cochlea, sometimes called the snail shell, that's concerned with hearing. It has a cochlear duct, and that's going to be kind of consistent with a vestibular membrane on its roof, and then on its floor it's going to have a kind of basilar membrane which supports the organ of Corti. And the cochlear branch of nerve 8 runs from the organ of Corti to the brain. So that's what we see here. This area where we have 3 fluid-filled regions. So 1, 2, 3. And it's in this center one that we have this darker piece of tissue, and that's the organ of Corti. And of course, this goes throughout the cochlea. Even though they've taken a cross-section, it looks like there's many different areas that contain the organ of Corti. It's actually more, if we kind of unravel our spiral, it's more like 3 chambers that are then rolled up. So the organ of Corti would be located here along kind of the bottom of that entire middle cochlear duct. And then from that, we would have the cochlear nerve, which is a division of the vestibular cochlear nerve. Here's just a better view. So just get a better view of the cochlear nerve, a division of cranial nerve 8. We can see how they've taken a cross section, but we're going to talk more about physiology when we see the kind of unwound version. So here we're looking at just one of those areas that have our 3 areas that contain fluid. So this is within the entire cochlea. In the middle layer is going to be our cochlear duct. The cochlear duct is going to contain endolymph, and it's going, just a type of fluid, and it's going to be outlined on its bottom by kind of a basilar membrane, on its top by a vestibular membrane. And then if we look at the organ of Corti itself, which is just a group of receptor cells and supporting cells, we find that right on top of the organ of Corti is the tectorial membrane. So again, we're focusing mainly on the cochlear duct, which is our middle region. The cochlear duct contains the organ of Corti. The organ of Corti sits atop the basilar membrane, and under the vestibular membrane and close to the organ of Corti is the tectorial membrane. Both the scalar vestibuli, which is this area up top, and the scala tympani, which is the area below, that contains perilymph, a different fluid. So endolymph is found in the cochlear duct, which is also called the scala media. Let's look at the organ of Corti in more detail. So here is our tectorial membrane right there. And then we have our basilar membrane down here that our organ of Corti is sitting on. We have our wonderful support cells supporting our receptor cells, or what's being called outer hair cells. So these cells would have to receive a stimulus, create a receptor potential, then that signal could travel to the cochlear nerve and then to the vestibulocochlear nerve. So this is what we're dealing with as far as our setup. So the outer hair cells are receptor cells. They contain stereocilia, which are kind of like regular cilia, but a little more sturdy. They are going to be communicating with the cochlear nerve, and they're supported by supporting cells. We see on top of the stereocilia is the membrane of the tectorial, and that if this tectorial membrane were to come down or this basilar membrane were to be pushed up, it would make the stereocilia hit the tectorial membrane. And if the stereocilia hit the tectorial membrane, it bends the hairs. It bends the stereocilia. And bending the stereocilia actually opens ion channels, which would cause sodium to rush into the cell. And that could cause a graded potential, which is in a separate cell, so we're calling it a receptor potential. And if that graded potential was strong enough, it could travel to these dendritic endings of the first order neuron called the cochlear nerve. And that would then be carried, if strong enough, to the brain. So let's talk about the properties of sound, and we're going to get back to the true physiology and step-by-step of auditory processing. So first and foremost, sound is a pressure disturbance that really is basically alternating areas of high and low pressure. The pressure disturbance forms a wave, but it's produced by a vibrating object. A sound wave moves outwards in all directions. It has an S shape. That's also called a sine wave. Properties of sound include frequency, wavelength, amplitude, pitch, and loudness. So a louder sound actually has higher amplitudes. So that's subjective to interpretation of sound intensity, but we do know when measured, loudness is going to have a higher amplitude. That's if something's more loud compared to something that's light or not as loud. Pitch is the perception of different frequencies. So higher the frequency, higher the pitch. And then amplitude, we're talking about the height of the crest of the waves, of each individual wave. Wavelength, we're talking about the distance between the top portions of the waves or two consecutive crests. And frequency is the number of waves that pass a given point in a given time. So if more waves pass a given point in a given time, that's going to increase pitch. So here is a sound wave traveling from the outer ear to the inner ear. Notice that as we go through the middle ear, we're going to amplify the sound waves. That occurs because of the auditory ossicles. That is their role. It's the amplification of sound waves. So here we have the pinna. From the pinna, we'll move to the auditory canal, also called the external acoustic meatus. Sound waves will then hit the eardrum. The eardrum will vibrate and cause the auditory ossicles to vibrate. The auditory ossicles contain the malleus, incus, and stapes. The stapes sits on the oval window, so when it vibrates, it pushes on the oval window, which moves the vibrations into the inner ear, and then those vibrations will travel through the fluids in the cochlear duct or through the cochlear canals. And if the sound was never loud enough to be heard anyways, it'll just die out in the cochlear canals. So let's look at the waves. We start out with some pretty reasonable auditory waves. They're going to keep the same amplitude. As soon as they hit the auditory ossicles, we see amplification. That amplification continues into the inner ear. Because there is fluid in the inner ear, but air in the middle and outer ear, we had to amplify the wave so that it could actually do stuff or move the fluid in the inner ear. It's a lot harder to move fluid than to move air. So that amplification continues until it dies out as it kind of leaves the cochlear canals. So let's look at the process step by step. So first and foremost, sound waves vibrate the tympanic membrane or the tympanum, or you can call it the eardrum, but sound is picked up from the ear. It travels via the auditory canal to your eardrum. So that's our step one. It vibrates the tympanum, which causes the auditory ossicles to vibrate. So that pressure is amplified. Pressure is amplified as it moves from the middle ear to the inner ear. What we're seeing here in blue is the unwound cochlea. And I would think of the cochlea in this way. It's easier to think of it in this way than it is to think of it in its wound shape. It's a little disorienting that way. Pressure waves will enter the scala vestibuli via the oval window. So we're seeing here that pressure waves created by the stapes pushing on the oval window move fluid that's in the scala vestibuli. So they move through that membrane, that bony membrane, and it'll move fluid in the scala vestibuli because we just amplified those waves. So that sound wave is going to move through the scala vestibuli, and if it's not loud enough, it would just continue moving till it goes around the bend called the helicotrema, and it'll eventually just kind of die out. It would move into the scala tympani. But if the sound wave is strong enough, this movement bumps into the cochlear duct. When this movement bumps into the cochlear duct, it will push the tectorial membrane into the stereocilia. So remember, that is the organ of Corti that exists within the cochlear duct, which is this middle part. So essentially what we're seeing here is sound waves, if loud enough, will bump into the cochlear duct and move the stereocilia into the tectorial membrane. And if we move the tectorial membrane and the stereocilia, it bends them, and we know when we bend them, we create a depolarization, which can be communicated to the cochlear nerve. So you can see here that sounds with frequencies below hearing travel all the way to the helicotrema, do not excite hair cells, but Sounds in the hearing range go through the cochlear duct, vibrating the basilar membrane and deflecting hairs on the inner hair cells. So we hit that basilar membrane, we push the cells up into the tectorial membrane, and that's all we wrote for bending of the stereocilia and creation of a depolarization signal that can travel via the cochlear duct to the central nervous system. Once we have the signal in the cochlear duct, it then travels up the brainstem, to the medulla oblongata. Once it reaches the medulla oblongata, it travels up to the midbrain, and then it'll synapse in the thalamus, because remember the thalamus is the relay station for the brain. And then from the thalamus, it's going to be conveyed to the auditory cortices in the temporal lobe, and that's the appropriate somatosensory cortex for sound. So here again is our basilar membrane. Sound waves that are strong enough send vibrations that push on the basilar membrane, and will push these stereocilia into the tectoral membrane. That will bend them, cause a receptor potential here that's communicated to the cochlear nerve, and that signal gets to travel via the vestibulocochlear nerve up the brainstem to the thalamus, then out to the appropriate cortice. Next, we're going to talk just more about auditory processing. So impulses from specific hair cells are going to be interpreted at specific pitches. Loudness, again, is detected by not only the number of action potentials that result when the hair cells experience larger deflections, but also on the amplitude of the signal that was detected. There is a localization of sound that depends on the intensity, but also the timing of the sound waves and how they reach both ears. So the fact that the sound wave reaches your right ear before your left would give you the idea that it's something closer to your left side, something closer to your right side than your left. So it's because we have two ears that we can localize sound. Let's look at some homeostatic imbalances of hearing. So we have conduction deafness and sensorineural deafness. Sensorineural deafness is when something's wrong with the cochlear hair cells or auditory cortical cells, or even the cochlear nerve or vestibular nerve. But we're going to see that gradual hair loss or something happening to the stereocilia is the general reason that sensorineural deafness occurs. Sensorineural deafness is deafness that occurs because the auditory signals can't leave the organocorti or the inner ear and be conveyed to the brain. Whereas conduction deafness is because of blocked sound or something that's blocking the conduction to the fluids of the internal ear. So this could be something as simple as impacted earwax, a perforated eardrum, inflammation of the middle ear, otosclerosis of the ossicles, where they kind of fuse together and can't amplify sound to send it to the inner ear. So there's a difference between conduction deafness and sensorineural deafness. Conduction deafness, something wrong with the conduction of sound waves to the inner ear, whereas sensorineural deafness is where we can conduct the sound waves to the inner ear, but they can't make it from the inner ear to the brain. All right, we're going to move on to a new topic next. We're going to talk about nerve structure. So we've been talking about nerves, cochlear nerve, different cranial nerves. We've been talking about the peripheral nervous system and how it contains nerves, but we haven't defined nerves, not strictly. So a nerve is a bundle of neuron axons or fibers, if we're talking the peripheral nervous system. It's cord-like, it's kind of like a muscle, especially skeletal muscle, if we look at the anatomy. So we'll see similar terms just with a different suffix. Again, very cord-like, it's of the peripheral nervous system. We're going to see bundles of axons, each with their own connective tissue called an endoneurium. When they're bundled together, that bundle of axons is called a fascicle. Fascicles are going to be covered by a connective tissue covering called the perineurium, and bundles of fascicles will be covered by a connective tissue covering called the epineurium. So again, very similar to the anatomy of muscle, but instead we're looking at different cellular structures having this bundles upon bundles appearance. So again, the connective tissue coverings for a nerve are the endoneurium, perineurium, and epineurium. The endoneurium is loose connective tissue that surrounds or encloses axons. It'll also enclose or cover myelin sheaths on those axons. The perineurium will be more coarse as far as the connective tissue is concerned, and it is bundling fascicles. So it bundles fibers into groups. We're calling those fascicles, and the perineurium surrounds fascicles. The epineurium is tough and fibrous. It is a connective tissue sheath around an entire nerve or many fascicles. We also want to classify nerves just a little bit better. Most of this information should be review. But we have sensory nerves, motor nerves, and they can also be called afferent nerves or efferent nerves because sensory nerves would carry impulses only toward the central nervous system, and motor or efferent nerves would carry impulses only away from the central nervous system. So that you should be familiar with. But most nerves are actually a mixture. They're mixed nerves that carry both afferent or sensory information and efferent or motor information. And some contain both somatic and autonomic or visceral fibers. So a lot of times our peripheral nerves contain axons of many different types of neurons. A mixed nerve contains both sensory and motor fibers. Impulses, therefore, are both going to and leaving the central nervous system. There are 12 cranial nerves, and I do recommend that you read in your textbook about them. We're going to talk briefly about them. It will require a little bit of memorization on your part. So if you have the old textbook, these are the page numbers for the cranial nerve information. Should be pretty similar for the new textbook. So you can remember the cranial nerves in order by name with this acronym. So on occasion, our trusty truck acts funny. Very good vehicle anyhow, or you can make your own. I highly recommend that. And then there's also a phrase for remembering cranial nerves in order by function. So here they're using S, M, and B. B is for both sensory and motor functions to that nerve. S is for sensory only, and M is for motor only. So for example, you have olfactory as one of your sensory only cranial nerves, and that's going to match up with on olfactory, some only sensory. But again, that cranial nerve acronym is some say merry money, but my brother says bad business, merry money. Sorry, that's a tongue twister. So again, your first cranial nerve is your olfactory nerve, and that's going to be in your olfactory bulb. Then you have your optic nerve. Then there's oculomotor nerve. So those first three have O's: olfactory, optic, oculomotor. Then I'm just going to go through so you can hear their names. We have the trochlear, trigeminal, abducens, facial nerve, vestibulocochlear nerve, glossopharyngeal nerve, vagus nerve, accessory nerve, and the hypoglossal nerve. I highly recommend charts like this where you have a list of both the Roman numeral and the name for the cranial nerves 1 through 12. You have information on whether or not different ones are sensory or motor, and you have information about whether or not they are parasympathetic or part of parasympathetic nervous system and contain thus those fibers. Good examples to take from this particular slide here would be the fact that the olfactory and the optic nerve are going to be solely sensory. Both of them are solely sensory. Later on, we'll also see that we can group nerves based on their function. For example, the trochlear, oculomotor, and abducens nerve, they're going to be able to move the eye. Next, we'll briefly talk about the spinal nerves. You need to know there's 31 pairs of mixed nerves that are spinal nerves, named for their point of issue from the spinal cord. They supply all the body parts but the head and part of the neck. And there are 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal spinal nerve. Now, for most of these, we see that the number spinal nerve matches the number of vertebrae. Not the case for the cervical nerves. There's actually 8 of them, and that's because 7 of the cervical spinal nerves exit the vertebral canal superior to the vertebra for which they're named, but C7 actually has one, or a spinal nerve that exits the top and the bottom. So there's going to be one that exits the canal inferior to C7. So you get 8 spinal nerves for the cervical. And then other spinal nerves are just going to exit all inferior to their vertebrae for which they are named. So here we can just see a basic overview. So C1 through C8 for the cervical nerves, T1 through T12 for the thoracic nerves, and we're just correlating them to their vertebra. And then lumbar nerves is L1 to L5, sacral nerves S1 to S5, and then there's just one for coccygeal, but it actually gets C sub 0. And then our last topic is going to be reflexes. So make sure you know the definitions: inborn or intrinsic reflex, and learned or acquired reflex. So an inborn or intrinsic reflex is one that is predictable. It has a motor response to a stimulus that you naturally developed. It's rapid, it's involuntary. It may not be an immediate reflex that you develop. It takes babies a couple of months to develop some of these predictable motor responses, like maintaining posture, being able to control visceral activities. But by the time you're an adult, these are definitely inborn or intrinsic. They're not things you have to think about on a daily basis. Learned or acquired reflexes result from practice or repetition. For example, driving skills, riding a bike, playing the piano. And then we want to talk about reflexes more, and there's actually 5 parts to a reflex arc. So here, again, the numbers say 4, but here would be number 5. So components of a reflex arc: this is a neural pathway. We start with the receptor, which is where we're going to get the stimulus acting on the receptor. If it's a special sense receptor, this would initiate a receptor potential. If it was a general sense receptor, it would initiate a generator potential. But we'd get a graded potential that could be communicated to a sensory neuron. Graded potential can create an action potential that's strong enough. So our next step is sensory neuron. A sensory neuron will transmit a signal towards the central nervous system. Now, this could be a circuit that's monosynaptic or polysynaptic. So most sensory signals are going to be polysynaptic. Many ascending signals or pathways include 3 neurons, a first, second, and third order. The third order being the last part of the circuit level processing that brings the signal to the brain. In the brain or spinal cord, we could have integration. A lot of reflexes actually occur in the spinal cord, but it's our central nervous system that carries out integration, or kind of the perception level of processing. And then it's perception that decides on a response. That response is going to be communicated to an effector organ by a motor neuron that conducts efferent impulses or information away from the central nervous system or our integration center. Effector organs include muscles and glands. Muscles contract and glands secrete, but they're going to respond to whatever the efferent impulse is by producing their effect. So here's an image I would recommend you study. So we have the different parts of a reflex arc. They're showing the spinal cord because a lot of reflexes do not need to be communicated to the brain. They can actually be carried out by the interneurons or that type of integration of the spinal cord. So we have, for example, a receptor in the skin that's going to pick up on a sensory signal, be carried by this blue sensory neuron to the spinal cord. The blue neuron, which is a sensory neuron, will communicate that information to the green interneuron in the spinal cord. The green interneuron will carry out the perceptual level integration and then determine a response, which it will communicate to the neuronic synapses with here in red, which is a motor neuron. That motor neuron will carry the signal away from the spinal cord, which is central nervous system, to an effector, and here they're showing you muscle as an effector organ. It could also be a gland. So be aware of the 5 parts of a reflex arc. Be aware of learned or intrinsic reflexes. And we want to talk just kind of briefly here about spinal somatic reflexes and autonomic or visceral reflexes. So for spinal or somatic reflexes, the integration center is in the central nervous system. For most reflexes, that part of the central nervous system that is the integration center is the spinal cord. And so these effectors for a spinal somatic reflex would be skeletal muscle. Again, soma means body, so it's telling you in the name where the information is coming from and also what we want to act on, which is the body. The autonomic or visceral reflexes, so visceral is in the name, autonomic like automatic, so these are involuntarily controlled reflexes, and therefore they're also going to act on smooth muscle, cardiac muscle, or glands, which are automatically controlled effectors. Skeletal muscle is not automatically controlled, it's voluntary, and it's the somatic nervous system that actually controls the skeletal muscle. Again, autonomic visceral reflexes going to a lot of things like abdominal organs or just body viscera in general. And we can actually see that we can test, or there's different tests for spinal somatic reflexes and autonomic or visceral reflexes. And testing of somatic reflexes especially is important clinically to assess the condition of the nervous system. And that is it for chapter 13. So make sure to look over chapter 14 as well. But review parts 1 through 4 for chapter 13 and make sure you haven't missed anything.